Essential biological functions depend on a tightly regulated balance between cell proliferation and survival. In cancer, mutations often occur in genes that encode tyrosine kinase proteins, disrupting this delicate balance, producing perpetually surviving but dysfunctional cells. These mutated clones dominate the physiological environment with overwhelming numbers, consuming nutrients at the expense of biologically essential cell rivals, forcing these functionally superior but less fit cells into extinction. FMS-like tyrosine kinase 3, or FLT3, regulates pluripotent stem cell proliferation and is downregulated during hematopoietic differentiation. Internal tandem duplication, or ITD mutation, leads to disruption of a repressor sequence in the juxtamembrane region of FLT3, which in turn leads to constitutive activation of the receptor via autophosphorylation. This initiates a number of downstream survival pathways, such as phosphatidylinositol 3 kinase, PI3K, which blocks apoptosis through phosphorylation of the BCL2-associated death promoter BAD, and RAS, which activates proliferation effectors such as mitogen-activated protein kinase, MAPK, and extracellular signal regulator kinase, ERK. Inhibiting the constitutive activation of FLT3 is a promising target for managing FLT3-dependent leukemias. Janus kinase, or JAK, is an essential mediator of hematopoiesis, regulated extracellularly by cytokines and intracellularly by suppressors of cytokine signaling, or SOCs. A base pair substitution on codon 617 of the JAK2 gene leads to a somatic mutation that encodes a cytoplasmic tyrosine kinase that escapes regulation by SOCs, initiating STAT, which phosphorylates intracellular substrates and activates cell proliferation pathways such as PI3K and MAPK. This gain-of-function mutation is prevalent in myeloproliferative disorders such as polycythemia vera and essential thrombocythemia. In primary myelofibrosis, cytokines and other factors released from rapidly proliferating and apoptosing megakaryocytes result in bone marrow collagen deposition, disrupting the marrow architecture and reducing its capacity to produce adequate numbers of functional blood cells. Ineffective hematopoiesis ensues, followed by life-threatening bone marrow failure. Inhibiting the constitutive activation of JAK2 is a promising therapeutic avenue for managing myeloproliferative disorders. Angiogenesis is a complex process mediated by a series of interactions between proangiogenic and anti-angiogenic ligands and their receptors. Two distinct but complementary systems have been identified as vital to tumor-mediated angiogenesis. Vascular endothelial growth factor, VEGF, and its receptor mediate pathways essential in the early stages of angiogenesis, promoting proliferation, migration, and survival of activated vascular endothelial cells and capillary tube formation. Angiopoietins and Ti receptors play a critical role in mediating reciprocal interactions between the endothelium and surrounding matrix and mesenchyme and in blood vessel maturation and stability. Interdiction in the VEGF and angiopoietin Ti2 axes, which are non-redundant and complementary components regulating tumor angiogenesis and vascular maintenance, is a promising therapeutic avenue for managing the morbidity and mortality associated with many malignancies in combination with chemotherapy and molecular targeted agents. The ubiquitin proteasome system regulates signal transduction in tumors through targeted degradation of key proteins that prevent cell growth and survival. One such regulatory protein is nuclear factor kappa B, NF-kappa B, which becomes activated when its endogenous inhibitor, IκB, is tagged by ubiquitin and degraded by the proteasome. Activated NF-κB transcribes genes that promote cell cycle progression, angiogenesis, metastasis, and survival. Proteasome degradation of IκB, activating NF-κB, has been implicated in malignancies such as multiple myeloma and mantle cell lymphoma. Tumors are exquisitely sensitive to proteasome inhibition, making this protein degradation system a unique target for subverting essential processes of neoplastic disease. Chemicals and ionizing radiation cause damage to the DNA molecule that is repaired in cells by multiple cellular proteins. Poly-ADP ribose polymerase, or PARP, is a nuclear protein that assists in base excision repair of damaged DNA. PARP also induces chromatin relaxation, increasing accessibility of other repair enzymes to the site of DNA damage. Due to its role in DNA repair, PARP has also been implicated in the development of anti-tumor drug resistance. Targeting the PARP-mediated DNA repair pathway may potentiate the effects of chemotherapy and radiation therapy and reduce anti-tumor drug resistance. Specifically, PARP inhibition may be effective in malignancies where there is a defect in homologous base excision repair, such as BRCA1 and 2 breast and ovarian cancer. In anaplastic large cell lymphoma, a translocation between chromosomes 2 and 5 fuses anaplastic lymphoma kinase, ALK, to nucleophosmin, NPM, forming the NPM-ALK oncoprotein. In non-small cell lung cancers, this translocation fuses ALK with echinoderm microtubule-associated protein-like 4, EML4, forming the EML4-ALK oncoprotein. These fusion oncoproteins autophosphorylate the ALK tyrosine kinase receptor, constitutively activating downstream proliferation and survival pathways, RAS and ERK. Inhibiting the constitutive activation of the ALK kinase receptor is a potential therapeutic path forward for managing ALK fusion oncoprotein-dependent solid tumors and hematological malignancies. The MET oncogene encodes a transmembrane tyrosine kinase receptor activated by hepatocyte growth factor, or scatter factor. When scatter factor binds the c-MET receptor, downstream signaling pathways and effectors such as Rho, FAC, PI3K, ERM, and beta-catenin are activated, which mediate mitogenesis, motogenesis, morphogenesis, and angiogenesis. Specifically, the activation of c-MET facilitates tumor cell motility and invasion and promotes tumor growth and spread. The MET oncogene is also amplified in settings where resistance has evolved to drugs that inhibit mutated epidermal growth factor receptor. Prevention of c-MET-mediated processes provides an effective route for managing invasive tumors of high metastatic potential and for resensitizing EGFR-dependent tumors to therapies that target those constitutively activated receptors. Cephalon Oncology has established a proprietary portfolio of tyrosine kinase inhibitors, along with a recognized expertise in apoptosis and cell signaling, translating progress in identifying pathways that govern the cell processes leading to and perpetuating neoplastic disease into the practice of hematology and oncology. In doing so, we are realizing our promise to extend and enhance the lives of people with cancer through the discovery and development of innovative medicines that fulfill unmet patient needs.